Identification and clustering network of virulent Aeromonas Hydrophila, strain number C1613425, hypothetical proteins. Aeromonas hydrophila is a gram-negative mesophilic species ubiquitous in aquatic environments, which causes infections in multiple host species, including fish. The U.S. channel catfish industry affected by this species since 2009, and it caused extensive mortalities and economic losses to the channel catfish industry in U.S. What we did as a team, sequenced the complete genome of this species, strain number with this strain number, which was isolated from an outbreak in catfish from a commercial production pond in Mississippi. Many proteins from its genomes are not assigned a role. So this type of proteins are called hypothetical or putative proteins. And these proteins remain to be elucidated so that their function and their potential biological roles can be identified or assigned. For this species, actually 1,082 out of 4,879 proteins are unknown, hypothetical. So we just worked on this 1,082 unknown proteins. So we filtered those hypothetical proteins through a pipeline from a recent paper. It is in the references. PFM and catch databases are used to retain hypothetical proteins at their consensus. In other words, if they hit both databases, we retain the hypothetical protein sequences. So it was a harsh filtering which left only 83 sequences, and these 83 sequences were submitted to BLAST, DEG and PSORT databases to obtain more information about homolog sequences, essential genes, and subcellular localization. Results from these three databases are summarized as a single table, and here I can show the top 5 entries of this table just to give an idea of what type of data we used to construct the network. So here's the table which shows the merged information from these three databases. So we see number of blast counts, average blast score, and PSORT domains such as unknown, cytoplasmic, periplasmic, and so on. And also we have DEG related features such as eukaryote, archaea, and bacteria. So we used these as the features to generate or to construct distance metrics from. So the table columns are treated as features, and they are employed to construct a weighted similarity network of relationship between the remaining hypothetical proteins. Normalized go-over distance is used, and we used an R package, cluster package, to compute the distance, after which the distance is converted to a similarity using the relationship. Since it is normalized, it is between 0 and 1, we just convert it to similarity using 1 minus the distance between hypothetical proteins. Similarity scores applied a threshold to be able to end up with somewhat sparse network, otherwise we cannot make a meaningful analysis. And then we applied a proportional to the based on the centrality of the nodes. String database reported connections between hypothetical proteins in each cluster. For example, SEC-ID2 from green cluster, SEC-ID3 from blue, and SEC-ID4 from red. So relationship between these three distinct hypothetical proteins and a protein named AHV, which is classified or determined as RTX toxin protein from Aeromonas Hydrophila strain YL17. So these three distinct hypothetical proteins from distinct clusters has relationship with this RTX toxin protein. So this was interesting for us to see. And you can see the hypothetical proteins with sequence IDs SEC-D, D4, D3, and D2, and relationship with this RTX toxin protein. So this research just aimed to analyze hypothetical proteins using network representation, and it provided us distinctive results about potential toxin proteins to investigate further. Thank you for listening.